I'm Carol Spencer. I'm the staff curator for amphibians and reptiles, or staff curator of Herpetology. We have about almost 300,000 specimens, so we're one of the very large collections in the United States and the world, especially one associated with a museum and a university. A lot of our specimens are actually collected for research purposes. So for example, I have that big jar of the slender salamanders that I showed people, the trichiceps, and those were collected over many years, over probably 80 different years, from different localities, and really they show a breadth of sizes of those animals. They might not look different to us, but somebody who does a lot of research on them, they look different. They show a breadth of kinds of things that those animals ate. You can do stable isotope research on those to look at what changes in their diet over time. There might be morphological differences within the populations, especially in some color pattern and things like that. And so really the idea is that these are a resource for everyone to use in the future. And a really good example of why we collect so many animals is the chytrid studies. So people were looking at this fungus that occurs in these amphibians, but we didn't really know why it was, why it got introduced here, if it was introduced, why it was all over the place, and we didn't know what year it had shown up. And so by being able to swab specimens that have been collected over 150 years here in the United States, you can actually try to look and see what year it showed up in different areas and sort of track its progress. So those kind of studies are really important, and really important especially considering we have a lot of climate change, changes in our environment going on, changes in habitat use, and so that's why we try to get a variety of animals. So I think there's really a lot of different preparations we also do. As I was saying, we have a really big tissue collection. I think altogether it's almost over 45,000 tissues of amphibians and reptiles that have been collected since the 70s. And so those are all things that people can use not only for doing genomics, but other kinds of research where they need to have DNA, but it's more difficult to get that DNA from the specimens because they've been preserved in formalin. So, and typically it's kept in either like 70 or 100% or 95% ethanol. And depending on if it's formalin fixed first, then we would have also taken a tissue sample that was either frozen or kept in some other kind of liquid to keep it, preserve it forever. So, and so we want to give them a variety of tissues. Or people looking at gene expression in like the eyeballs or in the heart or the liver or the muscle. If people are doing that kinds of research, we actually collect a variety of tissues with each animal. And we also have these, the clear and stain collections, which we don't typically do as much. And again, it's sort of the same reason. It's trying to look at, it's another way to look at the animal by looking sort of at its skeletal components and its cartilage. I actually, because I do also field work on venomous snakes, another kind of collection we do is venom. So venom can either be frozen or dried. You can't put any liquids in it because venom is made out of basically all these different kinds of anticoagulants and enzymes that are produced by their salivary glands of the animals. This giant cocktail is used to basically, kill other animals and maybe partly digest them. So that's another thing that though we do collect and we do preserve in our tissue samples. A lot of what we do is trying to preserve things that we don't really know that somebody might be able to use later. Like you haven't even thought of this idea, but I mean, at the time that somebody collected that big giant lachesis, the big bushmaster, probably that person never thought that you could maybe study the venom or the venom glands, or you could study what the diet was later, or you could do genomics, like you can get the whole sequence of that entire animal. But that person took tissue. You can't get the venom once it's been in formalin, but you can go and collect venom in new specimens and try to really compare those things to things we've already collected. And some of the venom can be determined by the DNA of the tissue samples that have been already collected. You can look at the gene expression. And so all those samples are really important in trying to help determine how venom is evolving, how the snakes are evolving, and then also how people can sort of make new kinds of antivenom to help keep people alive. So again, we're really trying to give people as much data as possible. We make all the data immediately available online for any species, and so people can then use these for looking at species modeling with climate. And so it's really a lot of information that we try to keep for each animal that died or was killed. We have specimens from Darwin, we have specimens from Linnaeus. People collected these things more than 200 years ago, and they're still in collections in jars and doing great. So as long as you keep the fluid levels up, they'll just be preserved forever. So.